Hi everybody, this is my second video recording to walk you through the lab that you're going to be working on this week in lab. So with this lab, you guys are going to need this lab manual, the very last one, and you're going to be working on the first lab in that manual, the DNA fingerprinting lab. And so I'm going to share my screen with you now and kind of walk you through what you should be doing this week. All right, so Unit 4, Lab 1, DNA fingerprinting is where we are. And I know that you haven't really jumped into DNA too much in lecture yet, but you're getting there, so you're going to get some of this in lecture within the next week. But this should be something that you covered in high school biology. DNA fingerprinting, also known as gel electrophoresis. If you took 181, you ran a DNA fingerprint when you did the B speciation lab. This is the chambers, and you can see the chamber down here, the chamber that you have that agarose gel that's poured and solidified into that chamber. You use a micropipette to put some of the DNA into the wells of the gel, and then you hook it up to a battery and run electrical current through a buffer, which causes the DNA to move through the porous gel that you've created. With DNA fingerprinting, the reason why it's so important is because through this process, we're able to create a fingerprint, if you will, through the DNA that will help us identify people. So DNA fingerprints are used for things like when we're investigating who committed a crime. We can use it for that, which is what you're going to simulate through a paper lab this week. You can also use it for determining paternity. They also use it to look for when they try and determine who's selling, to target poachers in countries that are selling meat that's from endangered species, so they use it for that as well. So anyways, this is an example of what a DNA fingerprint might look like. You'll notice that the first column over here has a lot of lines, and those lines are going to basically be just a lane that represents different DNA fragment sizes. And so the size of the fragments are, you basically just count the base pairs. So the letters in the DNA, you count those, and that tells you how big it is. And so, for example, we have these numbers up here. So 1358 would be how many total base pairs are in that fragment that falls along that row. You'll notice that in a DNA fingerprint, if all of the DNA started up here at the top, in the top wells, you'll notice that different fragments have moved different lengths down the gel. And so I want to encourage you to use this DNA electrophoresis virtual lab here, because it's going to walk you through exactly why these DNA fragments, or it'll walk you through like how the fragments came to be. So we use something called restriction enzymes to cut the DNA to create the fragments. It'll also walk you through why different sized fragments move further and faster than other sizes. But walk through this electrophoresis virtual lab here, because what you would have done in lab, if you were in person, is you would have done 4 different activities that would have covered the purpose of restriction enzymes. It would have taught you how to use a micropipette, which some of you are already familiar with. It would have taught you the purpose of the restriction enzymes that cut the DNA and how to set up the electrophoresis unit. So because you're not getting that in lab, make sure that you do this virtual lab here so you can get that. And then the other thing for this lab today, or this week is activity 5 in the lab manual. It's asking you to analyze the gel once you've run the gel. So once you have the different patterns that you see here, it wants you to analyze those patterns. Well, that's kind of tough because we can't actually do the lab. So to make that work, you guys are actually going to do a paper lab. So you're going to simulate this with paper using scissors to cut strips of paper. And that assignment is this Who Stole the Crown Jewels assignment. All of the directions for that are here, but essentially you print this assignment out, you read the directions. It's going to tell you where to cut the DNA. You're going to be given crime scene DNA and then 4 different suspects. And based on where the restriction enzyme tells you to cut, you cut those strips of paper, you simulate gel electrophoresis, and then once that's done, you analyze that gel. So basically what you're doing is you're gonna look at which lane matches up with the crime scene lane. Once that is done, page 13 is where you would have drawn the gel if you had done this in lab. So I want you to sketch the paper lab outcome on page 13 of your lab manual. And then page 15 are your discussion questions for this lab. You can skip question 1 because since we didn't actually do it in lab, it's kind of difficult to answer that question. But questions 2 through 5, please answer those. Once that is done, that is all you have for this lab this week. So it should move pretty quickly. The other important thing, or the other important things this week is that this Wednesday, so April 1st, which is our actual lab day, at 11:59 p.m. that night, make sure that you have submitted the pictures of your cells and energy lab manual to your Google folder. I sent an email where I told you what page numbers need to be submitted to me. And please make sure that the file format is PDF because that is easiest for me to annotate on it and just quickly check, circle stuff that you're missing and give you a grade. The other thing that needs to be in the Google folder by 11:59 on April 1st is the energy lab report assignment, where you pick cell respiration or photosynthesis. And I've already talked to you guys quite a bit about this assignment, but that assignment sheet and exactly what I need to see on that assignment, all those details are in a yellow box on, actually I'll just show you. Unit 3, if you click on lab 1, you'll see the directions for that energy labs assignment here. So all of that is due April 1st. The next thing is, and this is coming up soon, but the next thing is, let me share something else with you. The next thing is you guys are going to be, and if you look in the lab schedule, you'll see this, but you guys are going to be doing DNA presentations. The way that we're going to do these DNA presentations is that you're going to you're going to do this in your group of 4, and you're going to create a Google Slides presentation where you all collaborate on this presentation. You all put in work to create the presentation. You're going to be assigned a topic, which I'm about to tell you what that is, and you're also assigned a target audience. And this target audience is you're going to present to this hypothetical target audience, assuming that they have very minimal knowledge of the topic that you're presenting on. The topics are either going to be a topic related to DNA fingerprinting or a topic related to bacterial transformation, which we'll cover next week. These presentations are on the details for the presentations are on the lab website, and I'll show you that in just a second. But they're 5 to 10 minute presentations. They have to be within that time frame or else you lose points. And what you're going to do is create the Google Slides presentation, and then you're going to schedule a time between the group of 4 of you guys. You'll schedule a time between the 4 of you to do a Zoom meeting just with you 4. You'll record that Zoom meeting where you're sharing your screen to walk through the PowerPoint presentation or Google Slides presentation, and you'll save that recording of the presentation, and then you'll share that recording from Zoom with me in your Google folder. So instead of you having to actually like present it live, you have the opportunity to just record it through Zoom between the 4 of you. And if you don't like that recording, you can record it again, and you can record it again. So you can do it as many times as you want until you're happy with it. That video recording has to be shared with me, and the due date for that is going to be April 15th. So you still have some time, but I wanted to go ahead and give you the instructions for this. So topics and target audience. So first group, Salim, Elizabeth, O, Julia, and Gemma. Your topic, you can see here, describe the use of DNA fingerprinting as a biotechnology tool to identify individuals and to show whether individuals or animals are related to each other. Your target audience is going to be a news interview with an expert panel. My next group, Jenny, Sarah, A, Emma, L, and Skylar. Your topic, describe the concept of gene transformation, and your target audience is high school history teachers. Next group, Maria, Diana, Karen, and Pavitra. Your topic is describe how using gene transformation can or is being used to solve a current health problem. Your target audience is our 12th grade science class. Can't talk today. Next group, Elizabeth G, Emma P, Ashley, and Jenna. Topic, propose and justify an experiment using gene transformation to solve a current environmental or industrial problem. And your target audience is a middle school science Olympiad student or students. Next group, April, Sarah K, Andrew, and Omid. Topic, describe the structure and function of DNA, and your target audience, your target audience is PBS informational programming or a documentary, something that would be seen in a documentary. And last group, Austin, Foster, Atia, and Noah. Your topic is describe the procedure of agarose gel electrophoresis, and this is, agarose gel electrophoresis is the same thing that you guys are looking at when you do this activity for lab this week. DNA digestion with restriction enzymes and its application to DNA separation. Your target audience is a tour group at the Museum of Life Sciences. So you can rewind the video to see your group's topic and target audience again if you need to. All right. So let me show you, let me stop sharing this and show you the screen again. I'm just going to show you the lab website. All right, so the details for this assignment, you go to Unit 4. The details for this assignment are going to be here. This should be clickable. I think they're going to make this yellow box clickable soon because they're going to add this in here. But the DNA presentation expectations, if you open that up, it's going to tell you exactly what needs to go into your presentation. So here is going to tell you what needs to go into the presentation. And then another thing that's going to be happening, and this is even later on in the semester, but you're gonna watch your recording back from your presentation, and you're gonna do a self-assessment based on what you see from your presentation. And the purpose of this is just for us to see if we can identify some things that we can improve on, because ultimately you are going to be doing another one of these presentations for your bean beetle project, and the goal is to if you do things, which I'm bad at this, if you do things like use filler words like or if you have some sort of distracting behavior, like you're twirling your hair or something the whole time you're talking, identify that and don't do that during the bean beetle presentation. So you will eventually do this self-assessment just to give you an idea for when this is due. This is the self-assessment, and this is just an easy grade. As long as you do it, you get credit for it. Self-assessment is due on April 22nd. So this is going to be coming before you do. This is going to be due before the bean beetle presentation. Let me see if there's anything else. Oh, and the DNA presentation, this is how you're going to get graded based on that presentation. So this is the rubric that I'll be using when I watch your video, if you want to refer to that. And notice there are points for your presentation time frame. So make sure it's between 5 and 10 minutes. All right. Last thing, your statistics assignment, I'm in the process of grading that right now. And so just make sure to look back in there on that assignment within the next week or so, and you should have a grade for that assignment. Let me know if you have any questions on that. It's important that you understand that basic concept of the t-test and the p-values and statistical significance, because when you write your bean beetle lab report, you are going to need to do some form of statistical analysis. And so most of you will just do a simple t-test for that. So make sure that you understand that stuff. If you have any questions about that assignment, reach out to me, especially before you write that lab report. I think that's it. You can go ahead and start working on this lab this week. As soon as you've watched this video, feel free to start on it. Like I said, it shouldn't take that long. I think most of you, it seemed like last week, you're trying to set aside time during the normal lab time frame on Wednesdays to complete the lab, and I think that's fantastic. For those of you that are doing that, keep doing that. That works out great. And remember, I am available on Wednesday. I'll send out a Zoom meeting link for you from 1 to 2:30. I'll be available to help you if you stumble upon anything and you need help with anything that you're working on. That's it. I hope you guys have a great rest of your week. I hope that you are successful on your last Bio 183 lecture exam. Thank goodness you get to use notes this time, but good luck with that. Let me know if you have any questions as you begin studying for that exam. All right. Thanks, guys.